Now it's sharing?
Yes, yes.
Okay, great. So that's it. So I don't know what is happening. Anyhow, for those who do not know about Porphyromonas gingivalis, this is a small reminder. It is the human oral pathogens is responsible for periodontitis. It's one of the other pathogens, and actually the pretty nasty one, because if you don't take care of it, then you might have a tooth loss and other systemic problems, including heart and kidney problems. There is no commercial vaccine available. I am being funded for development of a vaccine candidate from the Polish government, so let's see how it goes. We know that the pathogen hates oxygen, so essentially you have to grow it under anaerobic conditions. It is known also experimentally that it produces 3 types of the lipopolysaccharide, and yeah, there are 3 types of antigen, an ionic type of antigen and the Ktype antigen, which is commonly known as the capsular one. The Oldtype antigen is pretty much known. A lot of work has been done on it. We know its role, biological role. It protects the host from the environment, and it is also important for the structural support of the cell. So simply, if you don't have it, the bacteria are not very stable. And structurally, we know what is the structure that has been published and accepted generally. And genomic information is also fully described. So for the anionic type of antigen, we know that it is protecting the host, okay? Regarding the structure, essentially, there are 2 proposals what the structure might look like. Each group is saying, that this is the best, and frankly, there is no agreement, so we cannot say it is known, the structure. And the genomic information is partially described. Not all the genes are known, and not all the regulations are worked out. For the capsular type of antigen, it is very similar in theory, though, to the E. coli system. And structural information is not known because we simply do not know the isolated structures of the antigen. Nobody has done it, and people are simply waiting for somebody to do it. And genomic information, there is one locus sequence. It was for W50 strain, I believe, and function of the genes is unknown. And some of them can be predicted informatically, and I will be talking about it later on. So let's go to the Oldtype antigen. There are essentially 3 types of pathways, assembly pathways. They are differentiated by the type of enzyme which is used to flip the antigen assembled across the membrane, and that is the flipase dependent. That's WZX, WZY. The other one is ABC transporter dependent, which is dependent on the ATP hydrolysis to transport it across membrane. And the third one is the synthase dependent. We are not going to talk about it. There are essentially ways to figure out, what the biosynthetic pathways might look like, but not only that you can get the genomic information and essentially plug it in the computer, but you have to correlate it with experimental information. So if you have the structure and you know what is the function of each particular protein, that means you know which reactions can you assign to it, then you can plug it in the holes literally using many software packages. In this case, we used Pathway Tool software from Biosynthetic, and we actually reconstructed in silico the pathway of how it's synthesized and correlated, of course, with the experimental data. What you see here, everything is correlated with experimental data. So let's start with the UDP alpha D galactose, and it has the lipophilic part attached to it. by the enzyme WBaq, and once it sits in the membrane, then the other parts are attached. This is the rhamnose, then the other is glucose and galactose again. And this is the basic tetramer, okay, unit. And this is transported across the membrane by the flippase. This is WZX. And the enzyme is probably different than the one for the capsular antigen because the sequence similarity is different. Okay, so it's just too high sequence similarity. So we think that there are two different enzymes. And once it's transported into periplasmic space, then in that space, essentially there is the O antigen ligase, polymerase, which will assemble those parts. That's a bit like PCR, but this case is carbohydrate units. We know experimentally that there are 3 to 5 such units. In this case, we show it for 3 units, 1, 2, 3. But as I said, 3 to 5 units are known experimentally. So that part, okay, once it's made, has to be attached to lipid A, and lipid A is made by a different pathway. And this is first one has to synthesize so-called lipid 4A, then it has to add capped core, and then we have to add the KDO part to the lipid 4A. So again, we have reconstructed those parts. I hate it because, okay. And in this pathway, essentially lipid 4A biosynthesis, there are 2 parallel pathways which converge later. So they both start with UDP and acetyl alpha D glucosamine. And there are enzymes, despite the fact that they look the same, I mean, they have the same numbers, EC number, they are having different amino acid sequence, and they are different enzymes. Has been known as kind of a quirks of the Porphyromonas gingivalis, but at the same time, it allows it to make different versions of the antigen, okay, later on. So there are 2 parallel pathways, they converge later on, where we have the tetraacylated, okay, disaccharide. So in this case, this is tetra, but it's known that it can be from 0 to 5 acylation units. So frankly, this is a little bit more. The last step in here is the kinase, which attaches the phosphate group. That one is particularly nasty in terms of toxicity, because if you remove it, then frankly, the LPS, it's losing part of this toxicity. So we have it. Yeah, this is the other one. So once you make this basic unit, you have to attach it to the other part, lipid 4A, okay, and that one is attached by O antigen ligase. As I said, okay, it's probably different than the one from the capsular antigen. So once it's attached, the whole part sits in the membrane, and this part, okay, which is variable, is sitting outside, is most likely recognized by antibodies, and it's very poor recognition. That's why it's some different. Yeah. What about the A-type antigen? Essentially, there are 2 models. One is the Paramonov's model. Okay, this is the British group which published it, and the other one is Japanese group by Shoji. And the Paramonov's model essentially is polymerized mannose and branched, okay, and they base the structure on a really thorough LCMS and NMR investigation. So I think they have good case, you know, to support it. And the Japanese group based mainly on the genetic information. So in their case, the structure of the anionic type of antigen, okay, is very similar to what we've seen for the Oldtype, because it doesn't have the branch, and of course this one will have to be galactose to be like in the O antigen, but it's not known. So frankly, genetic information, okay, and the potential reconstruction. So as you can see, there are two incompatible structures. We don't know why it is such a big difference, but as I say, each group thinks, you know, that they are the best, okay, and we have to wait till they decide who has the right, or somebody else will come up with the different idea and decide maybe there is something in between, or maybe, you know, there is an error. So the Ktype antigen, essentially, it is a cluster of genes which was sequenced, and there are 13 genes. That was for W50, I believe, originally done. And each one of them, almost each one except the two, can be assigned a specific function based on the bioinformatic analysis. So for example, this first one, okay, can be assigned probably the first step in the reaction, which is the attachment of undecaprynyl phosphate to the sugar moiety, and it's the N-acetylglucosamine, and very similar to the WBaq enzyme as for the O antigen. The last one, we know that we can get the structure modeled as well, and it's probably important for the conversion from N-acetylglucosamine to N-acetylmonosamine. So yeah, that's it. Okay, so when we look at essentially known sequences, complete genomes, not partial, but complete genomes, and when compare the clusters which are responsible for the Oldtype antigen genes, okay, and proteins which synthesize them, then we see that they are very much conserved, okay, from here to here, that's everything the same, except those four, okay, genes. And those genes, when you look at the amino acid sequence, then we see that those genes most likely code for a chaperone. We frankly don't know what kind of a chaperone it is, but and what is the function. So we think that the fact that it might be the chaperone, maybe it makes a little bit of a different, okay, version of the Oldtype antigen, but we don't have the experimental evidence to support it. So the Vim cluster genes, this is another cluster which is taking part in the A antigen biosynthesis according to the Church's model, and part of those genes is important as well for the O antigen. Okay, and so this part is very much conserved. There is not much of a difference from here up to here. Okay, it's pretty much the same uniform. And the Ktype antigen cluster, it's a little bit more difficult here to see, but essentially we see that there are 13 genes, and out of those 13, 6 are absolutely conserved. This is the first two, okay, and the last 4, 1, 2, 3, 4. So essentially, wow, kind of walking through this end of the cluster, and this is the beginning, and what is in between, it's not very much conserved. Sometimes it's okay, sometimes there are big gaps and inserts which we don't know about. So that's the other part. Okay, so essentially, based on the information which we have, we can do something else. We can essentially take the amino acid sequence and see how close it is to the amino acid sequence of other enzymes, and then try to build a model and figure out, okay, how it might interact. So in this case, we did it for the first enzyme of the O antigen biosynthesis, this WBaq enzyme, and it was based on the other structures which was solved. And this is putative substrate, okay, docked in the active site. This enzyme normally should sit in the membrane. So this is the blue one, okay, part which we'll see. It will be kind of like a V shaped, okay, and it will be sitting in the membrane, pretty much like here. And we think that the lipophilic part, okay, which is attached here, will be somewhere in here. There are two catalytic residues. This is glutamate 93 and the aspartate 92. Oh, I'm sorry. Okay, and there is the catalytic magnesium, the part of the substrate we see it's right there. Okay, I cannot see it too well. So essentially, this is we are pretty confident about the model, but the reality, you know, might play tricks. So we have to wait till the structure is shown. The other part is the epimerase, which is essentially converting UDP and acetylglucosamine to N-UDP and acetyl mannose-amine. Okay, this is the enzyme which is at the end of the cluster of the K antigen. So again, we could build the model. We are pretty confident about the model. And this is the substrate docked in the putative active site. Okay, catalytic residue is right here. The reaction goes by in two steps. First one, this one attacks this one, and essentially it's arginine, I believe, 11. And then there is anti-elimination, okay, and essentially we have a reversal of the configuration, okay, on one of the atoms, and we essentially change the type of a sugar in here. So it's pretty simple, okay, but how it works, it's a little bit more complicated. So at the end, we would like to thank the people who are involved in helping with the project. This is the IT core of the Polish Academy of Science. Of course, the Schrödinger software for enabling us to use it, and specifically for working out the quirks, how to set up certain parts, okay, that will work on our system. Markus Krummenacker from SRI for the ortholog computation. And there's Ron Caspi for practically using the E. coli model to reconstruct the LPS part biosynthesis. And of course, the funders we would like to thank. This is the National Science Center of Poland, Opus grant. It was awarded to me. And the other one is the National Institute of Health, okay, awarding grant to Ron Caspi for his work on the in silico reconstruction of biosynthetic different pathways. So the stage where we have the antigen candidate ready to be tested in animals. I hope it will be tested in this year. And of course, you know, if there is something interesting, then we'll be happy to go to the IP office and say, you know, can you patent it? If not, then we'll just simply publish it. Any other questions?